inner membrane space. So if I have low hydrogen concentration in matrix, what does it translate into pH?
Basic.
High pH or low pH?
High pH.
High pH. So therefore we have, write the first one, pH goes up. ATP synthase pumps proton by active transport. One of the best way to study this one, not only find the right answer, you have to come up with an example, a case why the other answer is wrong. If you can do that, you're really mastering your test technique, because it's not just, oh, I find the right answer. It's, you know why the other one is wrong and you can fix them. So ATP synthase pumps proton by active transport. What is wrong in this statement?
It pumps by the proton gradient, not by active transport.
Yeah, it's a proton gradient. Thank you very much.
It's already sugar inside.
You're so kind.
Yes.
I really appreciate it. Thank you. Thank you very much. Okay, and the next one is called electrons gain free energy. No, electrons are source of energy. You push them, carry them from one to another. The cytochromes phosphorylate ADP to form ATP. The cytochromes, they don't do that. ATPase does that. And NAD plus is oxidized. What is wrong with this?
It's reduced, not oxidized.
NAD plus is already oxidized. You can't put extra charge in that, so that's wrong. 7, cells do not catalyze carbon dioxide because it's double bond or too stable to be broken. CO2 has fewer bonding electrons than other organic molecules. So it is stable, but it doesn't mean that it could be broken. It cannot be broken. CO2 is already completely reduced, and CO2 is completely oxidized. The molecule has too few bonds. So what is 7? If it says CO2, I have to be honest, when I took this last night, I was a little bit confused, because I thought if it's a CO2, I look at the oxidation state of carbon. So what would oxidation number of carbon be? You say carbon, oxygen is -2 times 2, -4 is equal 0, so carbon must be +4, right? Oh, it is true. So two ways you can say this is already oxidized. One is carbon oxidation state is +4. Remember, +4 to -4. This is highly oxidized. Or even if you don't do that, by looking at this structure, has lots of oxygen. 4 oxygen means this is just saturated with oxygen. So it's because carbon is already completely oxidized. And the last one, second to last. Which of the following is a true distinction between fermentation and cellular respiration? Only respiration oxidizes glucose. Only respiration oxidizes glucose. So you lose some molecules. So that could happen in the first part. The next one, NADH is oxidized by electron transport chain in respiration only. Remember, NADH is already reduced. We take it to mitochondria to oxidize it. Fermentation by non-respiration is example of a catabolic pathway. They're both examples of catabolic pathway. Substrate level phosphorylation, this is happening. Glycolysis is unique to fermentation. No, it is unique to glycolysis. And NAD plus functions as an oxidizing agent only in respiration. So oxidizing agent means gets reduced. Yes, this can get reduced, but not in respiration. You could have that in other part of the body that you use NAD+, like in amino acids, nicotine adenine dinucleotide. So that makes this statement wrong. So the correct answer is B. The last one, most CO2 from catabolism is released during. So what do you think? Which part of the cycle when we had lots of CO2? This one, we didn't get any CO2, we just broke it down into 2. Pyruvate to acetyl-CoA, we lose 1 CO2. This is the part that we lose the CO2, we made NADH, CO2 NADH. So from 6 to 4, we lost 2, and here we lost only 1. So this must be it. So it has to be in citric acid cycle. Right? All right, so I did send you this one, and if you check your email again later on, you will see some tests that I sent you. That's mostly about cell cycle. Hopefully, I'll get to this, and then I will provide you some tests and practice problem with this one. Please take them, and then if you have any questions, please email them. We don't have teacher's solution yet. So what I do every night, I'll have to take the test and see what is the answers, if I get it drawing right, and then I know all the answers. So you let me know if you have any question, and then I'll check with the test I've taken, and I'll provide you the answers. One other thing. Yesterday we were talking about enzymes. One other thing I want to point out, I've seen this quite often, that some students, they get confused to distinguishing between cofactor and coenzymes, right? So this is, I kind of dug deeper a bit and found as much as I could. So this is the enzyme. Imagine this part is a non-active site, and this is the active site. Substrate binds to the active site. So the category, if you break it down, this is, I want you to think of it this way. Cofactors are divided into coenzymes and metal ion. Okay, so coenzymes and metal ions. The coenzymes are non-protein organic complex that causes activator, right? So coenzymes are non-protein organic molecule that cause activation, and the coenzymes, they happen to bind to the active site, another part of active site. So the whole thing, this region, bind to a portion or different to a portion, to a segment, I guess, to a segment of active site. Okay. What are the examples? If they are non-protein organic complex, they have to have carbon in it, so NAD or NAD plus, FAD, coenzyme A, and vitamins are also coenzymes. That's why we eat vitamins. Metal ion, on the other hand, are bind enzyme and substrate together, so they act like a bridge. They bridge them together. So cofactors here, they bind metal ions and substrate together, so they bind to the non-active site, and then these are the examples. But metal ions, they do two functions. One is combined with protein as a catalyst. They come as a catalyst sometimes in some reactions, and also they bind enzyme substrate together. These are a couple of examples. So Fe plus 2, Fe plus 3, they are cytochrome and hemoglobin. That's why they have heme group in blood. Zinc is an alcohol dehydrogenase, and also works in carbonic anhydrase. We get to this in blood. And pyruvate phosphofructokinase that uses magnesium and potassium. So it should be clear what is the distinction between coenzymes and cofactor. So cofactors are very broad, and then divided coenzymes and metal ions. Usually, sometimes I've seen it in books, they say coenzymes, cofactor, they separate them in a sense that every time they say cofactor, they think about metal ion. So you can even classify them in two different groups. You can even say enzymes and cofactors side by side. So this is a metal ion, and this is a non-protein complex. They do that, but categorically, this is subdivision of this. That's how some books they write. All right, so this is the part, and I will go. Last time we didn't get a chance to go over nucleic acid. I need to do nucleic acid. I think it's a good idea because now genetics is mainly based on nucleic acid. We will have other people coming to our class. One of them is on vacation, so William, I believe, will be here tomorrow. So, and there might be other people, but it's okay. We'll keep going forward, and I really want you to study hard and just push forward. Okay, nucleic acid. A flashback. What was the subunit of protein?
Amino acids.
subunit of carbohydrate,
Monosaccharides.
fats,
Fatty acids and cholesterol.
fatty acids and cholesterol. Nucleic acid, what is the subunit of nucleic acid? Nucleotide. Okay, nucleotide is subunit and is made of sugar, nitrogen base. The book, sometimes I hear they say base, but I want you to emphasize on the nitrogen base, sugar, nitrogen base, and phosphate. Okay, so sugar, nitrogen base, and phosphate. And this is how, and there are two nucleic acids. One is called DNA, the other one is called RNA. DNA and RNA. So DNA stands for deoxynucleic nucleic acid. This stands for ribonucleic acid, and the structure ribose is a five-member ring. This is the part that you attach this to a base, OH, and CH2, and then you get OP. The reason I draw this because I want to make a point. Did everybody write this? I want to just erase this part. I need this. Can I erase this?
Yeah.
Okay. So this one, and then OP. Okay. So this is the base. This is the sugar, and this is the phosphate. Phosphate, base, sugar, and phosphate. You don't need to know details, but I need to point some stuff out. So this is they call it carbon number 1, number 2, 3, 4, 5. 5 carbon in it, and in carbon number 2 you have OH. If you have an OH, this is called ribose. If I get this one, and if I draw this without OH or hydrogen, the same, everything here, base, CH2O phosphate. If I do this, this is called deoxyribose. Right? Deoxyribose. Now, what is the charge of DNA?
Negative.
Okay. I'll come back to this a little bit more. So if we pack DNA with some protein, with some amino acid, we pack it in a chromatin, what kind of amino acid do you think you need to put next to this DNA? So this is DNA, Kevin said it's negative, and I need to pack this DNA into some amino acid proteins. This is my DNA packing into here, and then have another one here, and pack DNA here. This is called chromatin. So made of DNA and some protein or amino acid. What kind of proteins do you expect to be in here? I'll give you some questions. Glutamic acid, alanine, lysine, alanine, tryptophan, okay, and then I have histidine. I'll give you a point. Think about charge. What is this?
Negative.
So if you want to pack them together, could it be negative?
That might be positive.
Okay. Why histidine?
Because why is one of those four,
Okay,
which
4.
are the basic one.
How would basic help to?
It would neutralize the charge.
What is the charge on basic?
Positive.
Did you see the two logical steps?
Okay.
So this is negative. If you pack it together, it cannot be negative, repel each other. It must be opposites like positive. Even if you don't know, we know that basic amino acids are positive, so you choose a basic one. And out of those basic, lysine, histidine, arginine, histidine is the subunit that you see in DNA. Okay, so this could be in some of these test takings, The tough questions, they have 1, 2, 3, 4 logical steps. The easy question, 2 logic, A plus B, then you have to have C. All right. I know with the pace I'm going, I'm not going to finish this, but let's just speed it up a little bit. Can you talk to me about the bases? What are those nitrogen bases?
Adenine, guanine, thymine, cytosine, and in RNA uracil.
Okay. Adenine, guanine. They also classify them as 2 different categories. They call them pyridine or purine or pyrimidine. Which one of them are purine? Which one of them are pyrimidine?
Thymine and cytosine are pyrimidine.
But what do you know about the purine? Are they 2 ring or 1 ring? So
One ring.
nitrogen base look like this. I'll just here. This is the one of them is here. This is one kind, and this is another kind. So they call them double ring or single ring. The mnemonic that I want you to use to memorize this is this. Purine, just say pure as gold, and pyrimidine, say cut the pie. Just think, I'm dyslexic, I don't know how to write pie. So pie, right? Or pie, here. Cut the pie. So purine as a gold. Purine are adenine and guanine. Pyrimidine are cytosine, uracil, thymine. Or if these 2 are purine, the other 3 must be pyrimidine. The way you memorize them, we are double bonded, double ring or single ring. Purine, how many of these bases are in purine category? 2 or 3? 2. So associate these 2. So if there are 2 of them, so each of them is a double bond, if it works. So the other one must be a single bond. Now, what is the nitrogen base pair? Cytosine base pairs with the guanine, and adenine base pairs with the thymine. So that's how they come together. Okay. And I have one note here. The way you get uracil, adenine, thymine, if you have thymine, if you have cytosine, if you do deamination of cytosine, you end up getting uracil. Okay. Now, a little bit more about that DNA structure. What do you know about the DNA structure? It's a double helix. That's I might need. So DNA structure. So versus RNA. DNA is a double bonded, double helix, and sometimes they say this is like a right-handed double helix. It turns to the right. So this is a helix. Whereas RNA is a single bond. You know the bases, we have an adenine, guanine, cytosine, thymine. This is an adenine, guanine, cytosine, uracil. That's for this. DNA is made of nucleus, but this is made of nucleus and we translated into this. We have three types of RNA. One is called tRNA, mRNA, and then let's say rRNA, mRNA, and tRNA. Where are these made and what are the functions? Where is the rRNA?
Ribosomes.
Where is it in the cell? It's called ribosomal RNA, but usually this is in the nucleolus, and this is the message to make ribosomes protein. Ribosomes. mRNA is called messenger RNA. You make it in the nucleus, you process it, and then you send it into cytoplasm, and it has a codon, which is the codon or genetic language for protein synthesis. Protein synthesis. tRNA is in the cytoplasm, and brings anticodon. Anticodon, or in parentheses, I want to say amino acid. It brings amino acids to ribosomes. All right. More about the DNA. If I do need this. More about the DNA structure and packing. I kind of alluded to the fact that it packs with the chromatin. This is about DNA packing in eukaryotes and prokaryotes. One thing I would recommend, especially for this test and both, since they're both in both organisms, every time you learn, try to make a comparison between them to see how they are related, packed in DNA in prokaryotes or in eukaryotes. So in eukaryotes, they found in octamer. What is octo? How many subunits? 8 subunits. 8 subunits. They found in octo, and then usually, as I said, so this is the structure. You have DNA comes, wraps around, and then goes to this structure. I write 4 because they are dimers, so it becomes 8, and then goes on in this 4 dimer of subunits. So, and this is called linker DNA. So this is called linker DNA, and these are the histone subunits. These are the histone, they call it histone subunit. They have histidine. DNA in prokaryotes, they use an enzyme called DNA gyrase. What is the function of DNA gyrase? There is a part that I go over them that they talk about all the different structures, like different enzymes. What gyrase does is coils DNA. So if this is a loose DNA, goes through gyrase active gyrase, and then just packs the DNA all together. What is enzyme? What is antagonistic to gyrase? What enzymes unpacks DNA? I want you to know 2 of them. One of them is helicase. I'll get to this in a moment. Helicase, and the other one is called topoisomerase. I'll tell you how they work. Topoisomerase. These they do unpacking. Let's just be specific in all of them. Helicase works at a site called replication fork. I'll get to this when I talk about the replication fork, and this unwinds DNA, so has a kind of unwinding effect. Topoisomerase works downstream from replication fork, and it releases twists, and so it loosens or cuts, unwind, releases, twists, cuts, and loosens DNA. If you have 2 ropes, this is a good example. If you have a rope and these fibers that are attached, if you pull it apart, you see down a bit further, you see it just gets so convoluted, gets so tension in it. So topoisomerase does that. Helicase works at the replication fork. As it opens, it just unwinds the DNA. So these 2, they have antagonistic factor against gyrase. Gyrase coils them together. This unwinds at replication fork. This unwinds or cuts, it releases the twist downstream, further down the replication fork. All right, so that's the DNA what I need to do. Now, let's go, so we will DNA, we talked about the protein structure. Somehow I have genetic mutation here. I want to go over genetic mutation. Before I walk you through genetic mutation, why don't we go over some general idea? The best way to get this one, as we get to transcription, translation, you might feel inundated and with all these different names and terminology. So it's just to make sure, try them in a flashcard or set them side by side and test yourself. Sometimes you've heard this a lot. They call it central dogma of biology. So they say going from DNA to RNA, and then we would next go to protein. Okay, what is this process called, DNA to RNA? Transcription or translation?
Transcription.
Transcription. Victoria? Is this transcription or translation?
Transcription.
Okay, RNA to protein? Translation. Or some teachers, they add this. They say going from DNA to DNA, this is called replication. Is there any way I go from RNA to DNA?
Yeah, and retroviruses.
Yeah.
And what is the enzyme?
Reverse transcriptase, or actually, not sure.
Reverse transcriptase. And this happens in, they call it retroviruses. If I do that, what do I create? Do I create DNA or no? I create something slightly different than DNA. Can you name that? You've heard that. I create something is called cDNA. So when you make DNA to RNA, you have intron and exon. So you slice the introns out. But if you have RNA, processed RNA, so I'm just going to say processed RNA means without introns. If you go back, if you do this going back to DNA, what you make cDNA, and this is called complementary DNA, means you make a DNA that has no, in other words, now this DNA is probably the most pure or useful in a sense. It doesn't have a region. The introns, there is a part of DNA is called junk DNA. You transcribe it, but you don't use it. It becomes intron and you cut it out. Means once you make the cDNA, it's pure in a sense that every sequence in this DNA is being transcribed and is useful. You use them to make it for a protein. So this part has a junk DNA and other DNA, but if you go reverse transcriptase, you make cDNA that doesn't have any untranscribed or unwanted region of DNA. That's one way to sometimes. They like to do this. They make it to RNA, reverse transcribe it into this, so they make it pure, is lighter, less kilodalton bases, and this. So that's another way to do this. Okay, so this is for this part. Transcription. Oh, by the way, so the DNA, this one has nucleotide. This one has nucleotide. And this is, the other one is just amino acids. So these are the subunits of this. Nucleotide, amino acids. How many type of amino acids? Again, I'll go over this. Well, here, I don't want to jump ahead. I'll get to translation when we go to this one. So I did talk to you about going from DNA to RNA and then processing it going here. Would you ever do anything to DNA? DNA is a genetic makeup, right? You do not want to touch it. If anything, if you do any changes to DNA, that results in mutation. There is one other way you can get mutation. Either during the base change, we will go over different mutation here, Or there is another one called non-disjunction. This is at chromosomal level. This is very obvious and is more devastating, deadly in a sense that if you lose a chromosome, gain a chromosome, Down syndrome, Turner syndrome, Klinefelter syndrome, the individuals usually they don't survive, or for the rest of their life they're altered. You can see them either they're sterile or they look different. Whereas if the mutation happens at the DNA level, you could be hemophilic, colorblinded, you could survive, or like sometimes the life expectancy is like up to 20 years, sometimes you live normal life, but you have some deficiency. So this is most in cellular level, metabolic activity alters. Non-disjunction, if you lose or gain chromosome, this happens through cell cycle, it's the entire cell, it just acts differently because the number of chromosomes are different. All right, so let's go over the gene mutation. You classify them as two types. Mutations in, again, DNA, right? In RNA is already been transcribed, you do this. First is called point mutation. And point mutation is the following. Number one is called, so point mutation, you change one base pair. Okay, so it's called, there are two kinds, called transition and transversion. So either is called transition or transversion. What is transition? This is the part that you need to know what are purines and pyrimidines. So purines were adenine and guanine, pyrimidines were cytosine, uracil, and thymine. So, well, we don't have uracil in DNA, so it's cytosine and thymine, right? So transition means in a base pair you have, let's say, A, G, C, C, G. This would be, let's say, becomes A, and instead of A, you could have G. You cannot have C and T. Why? Because this is purine and purine. That's what they're trying to say. A, G, G, C, G, G. So this is a purine. Transversion, on the other hand, is a different group of base pair, different base pair family. Base pair family. Again, so A, G, C, C, G. This becomes to A, and instead this becomes C, and then, or A matches with T. C, G, C, C, G. So this is a pyrimidine. And the one thing is obvious that A and G, they don't go together, so that could cause a problem because they don't match, but this one would match. I mean, sorry, it's not matching. This becomes G, right? I'll take it back. You don't match it. This is in one of them, and the other strand would also change to match. Now, the further, if you classify them, how would it affect them? So this is the general idea, and then you go into another different classification, given point mutation. It's called, let's start with this, silence mutation. Okay, so what is a silence mutation? So I have some examples here. So for silent mutation, you don't see no change in amino acid outcome, and the reason is because, let's say you do a change in mutation and ends with CGC, right? And then you change this into CGA. Once this, in other words, you've done change in DNA that causes this becomes an mRNA, and you make another change that you get this one, right? So this one is code for arginine, and this is also code for arginine, and these are the codon in mRNA. the reason they call it silent mutation because if you change this base pair to this one, they're both coding for the same amino acid. And that's why if you look at the codon tables, each amino acid, it has more than one codon to represent amino acid. That's number, let's call it number 1. Let's call this A. B, the next one is called missense mutation. Missense mutation. And for the missense mutation, so it changes amino acid, changes into a different amino acid, results into different amino acids. So for this one, let's say if I have CGC ends us with GGC, this is the arginine, and then becomes glycine. Some of these changes could be conservative in a sense that if arginine is, look, this is a basic amino acid, this is a glycine non-polar amino acid. It could be devastating because this amino acid has to be in a basic condition. Once you change it into this is an amino acid that is in a non-polar condition, so it won't really react with this. But sometimes if you change, let's say, alanine to glycine, they are both non-polar amino acids. So the condition in the cell is a non-polar region, non-polar environment, so probably you won't see adverse effect. But if you see effect that goes from arginine to glycine, that's a basic amino acid, goes to regular amino acid, the amino acid tends to fold and form different structures. The example for this is in a Huntington disease, that glutamic acid becomes alanine, or in a sickle cell anemia that causes the different amino acid devastating effect. So silent mutation, missense mutation, I'll show you a way to memorize this in a moment. And C, let's call it nonsense mutation. A changes happens in DNA that when you translate it into amino acid, you end up getting into, this is they call it premature stop codon. Do you know what are the 3 stop codons? There is a UAG, you are gone, you are away, you go away, UGA. Am I right? So it causes premature stop codon. If you end up changes DNA, causes getting this and with this structure in mRNA, that's called a silence nonsense mutation. So the way I have another category, I'll go in a moment. The way I memorize it is, if sometimes you talk to somebody and this person is just talking crazy and just nonsense, what do you tell them? You say, stop, you're being nonsense, right? Think of it this way. So nonsense means stop. Missense mutation, you miss something. You made a mistake, you missed it, right? You take a test, oh, I missed this because I made a mistake. There must be a mistake. Something is changing to differently. So that's a missense mutation. Silent mutation, you make a mistake, but doesn't affect anybody, so that's all right. So silent mutation is there is no change in amino acid. That's because they have more than one codon for each amino acid. Make sure to get this. So these are all examples of point mutation. They classify them as a transition or transversion. If they want to test you for this, just find out what kind of mRNA they get. If the same family is called transition. If it's from different families, it's called transversion. Now, sometimes this is a point mutation. There is another part. If you have a sequence of DNA, you could insert a gene in a nucleotide, base pair in it, or you could take it out, and that's called insertion or deletion, and which ends into frame shift. And there's one point at the end I want to make. So let's go over insertion, deletion, and let's see if any insertion or how many insertions you have to leads to a frame shift. Okay, so is called insertion or deletion. For this part, example, let's say if I have CGA, CCA, and ACG, and GCG. So this is code for arginine, this is proline, and this is thymine, and this is alanine. So if you take one of them out, everything will change it, right? So if I take this one, if I take this one becomes CG, let's say CG, and then delete this, CCA, everything will shift. Then you consider all this together, ACG, GCG. So there's a 3 base per each amino acid. So as you can see, it becomes devastating. A couple of, sometimes, I hope it doesn't happen to you, people take multiple choice, they put answer A to B, or 1 to A, and what have you, everything just gets scrambled. So that's a insertion, deletion, and if you add one thing, you get, let's say if I add it here, you get CGA, let's say CCA. If I add this, then the whole thing will change up. The question I have is this. So every amino acid is represented by 3 base pair, right? 3 different code, 3 different nitrogen bases here. What if I insert or delete 3 amino acid? Would that also be referred to as frame shift mutation? This is called frame shift. Frame shift mutation.
That'd be more of just a deletion of like double codon.
I saw that in one of the tests. If you insert a multiple of 3, you do change the sequence of amino acid, right? Let's say if I insert here another CCA. Let's say if I insert right here, I insert CC, let's say G. However this is. So if I insert this, then you introduce a new amino acid here that corresponds to this. You should know that when you talk about frame shift mutation, if you want to have this, consider this, it has to be inserted anything other than multiple of 3. So either 1, 2, 4, 5, 7, right? So you insert 1 and 2, but if you insert 3 amino acids, 3 base pair here, it will not be a frame, it is a frame, it shifts them, but they don't classify it as this because that doesn't alter, it introduces new amino acid or takes one amino acid out. It doesn't changes one amino acid to a different amino acid, right? You get the point. So if you say multiple of 3 insertion, amino base pair again, that would be classified as frame shift insertion. Are you okay for this? So make sure to the next part that we want to go is called DNA replication. All right. So before I go over this, there are a few things. Let's think about different possibilities and go over enzymes, and then we put everything together. And there are a few things that I really want you to notice. There are different categories, different hypotheses for this. One of them, they said it could be conservative. Conservative means if I have one DNA, let's say this is a DNA, if I unbind it, I end up getting this, and this is the new one. That was one hypothesis they came up. And the second one was, they said it's called dispersive. Dispersive is that if this is a DNA, this is the DNA model, it would look like this. The new DNA would be this way. We make off of this way. That's the idea of dispersive. And then later on, they came up with something that's called semi-conservative. Semi-conservative. And semi-conservative is if this is a DNA, the new branch would look like this. right? These are the three categories, theories that they came up when they started talking about DNA replication. So among this one, which one do you think is the valid one? Yeah, so it has to be a semi-conservative, and we'll see in a moment that DNA replication is semi-conservative. All right, now let's go over the enzymes. What is the first enzymes that I need to know? The first one is called DNA polymerase. The name says it all. It's an enzyme that polymerizes DNA. Means you make more DNA from one DNA. So the function is that catalyzes elongation, and reads template, and elongation happens 5' to 3'. There's one category you have 3' to 5', which I get to that. So 5' to 3' reads the template, and then reads the template, which direction? 3' to 5'. I'll point this out when I get to that. And then it needs 3' OH before catalyze. Okay, that's number one. Number two, it's called primase, and this provides RNA primase. A little bit counterintuitive. And if you're doing DNA, right? I'm replicating DNA. There is no think or thought of RNA. How on earth I have to use RNA primase? When you start replicating, the very first thing that it starts doing is RNA primase comes, it starts binding, and DNA polymerase binds to RNA primase and elongates. In other words, this is what you need to just start it off. It binds, it anchors the starting point, and then this comes and extends and elongates this. Make sure to know this RNA primase. Again, a little bit counterintuitive, but that does happen. And then, so RNA primase, and then is replaced by DNA polymerase. So if this is a DNA 5', 3', this is a 3', 5', and it's just as if this is a primer would come and bind, and then it goes this direction because you have to make, this is a 5', 3', so that's how it works. The next one is called single, this is a bit mouthful, it's called, they call it SSBP, which stands for single stranded binding protein. Single-stranded binding protein, and what it does is aligns and replicates DNA. So aligns and replicates DNA. The next one is called gyrase. What is the gyrase? We went over this. So gyrase, once you open it, once you duplicate it, it coils DNA. The other one that Victoria said was helicase. Unwinds DNA at replication fork. Okay, I'm glad that you're writing it, because once I write the structure, then you can tell me where they're coming. And if I may put the 6 here, topoisomerase. Since I already mentioned it, but since we want to put it in one place, it releases releases, twists, and loosens DNA downstream. Okay. Let me see whether okay. I'll explain this and then we'll make it the comparison between prokaryotes and eukaryotes. You guys are okay, right? All right, I'm just gonna. Let's draw. I'm gonna draw it like this instead of curve. Okay. As soon as I mark one of the end, you should do the rest, right? So let's see if we can do this together. So, okay, I'm gonna go that direction. So this is a 3'. And one other thing I said, DNA is anti-parallel, right? So if one goes 3', 5', the other one is 5', 3'. So as soon as I put this, you should start doing all the way to the end. All you need from me to tell you which side is a 3'. So if it's a 3', what is this side, Vivian?
5'.
What is this side?
2'.
What is this side?
5'.
Okay. I really want you to get this straight. Template is the same as antisense, and it's also, they call it anticodon. Okay, template, antisense, anticodon. And if it's the other one, there should be sense strand. They don't have, or template something. Sense strand or codon. Okay. I wrote something here, DNA polymerase. Based on this, which one of them is the template? Oh wait, no. The template is for transcription translation. We are in replication, right? So this is true. This is true perfectly, it's right. But for now, you only use this if you want to transcribe, right? Transcribe, you have to transcribe one of them. But all you're doing, you're duplicating, you're replicating. Are we clear? So use this again for transcription only. Okay, I'll mark this and then I'm going to erase it because it's not applicable. We'll get that to the template. But please mark it and don't confuse this, okay? Are we all right? So if that's the 3', 5', which direction I would start? Good. I'm glad I made that mistake, but then now we can clear it. If I'm replicating or duplicating DNA, I do it on both strands, right? I'll give you one. I said it's semi-conservative. This becomes matches with the new. This matches with the new one. So 3', 5', 3'. So the replication happens 5' to 3', right? So if this is a 3', 5', and this is a, so DNA helicase comes over here, one here, probably one here. This is a helicase. Opens this up. This is also called replication fork. Replication fork. And then RNA primer would come, and as this opens up, and if this is the 3', so 5', so it becomes 5' to 3'. Opens up and then puts this base pair here, single stranded base pair. First point primer, and then RNA polymerase would come along. This is DNA. DNA polymerase would come along to make this. So as this opens up, you start making this. So you make this, and then goes further on. You make another one here, and then so forth goes this direction. right? So primer DNA polymerase, you make this, and this is a 5' to 3' strand. How about in the other strand? This one, what happens, this is a 5', 3', but this opens this direction, right? And then you put, so 3', this becomes 3, so this is a 5', and 3' there. So the other part, you start making this, but then Okazaki creates a strand, it's called Okazaki fragment. Okazaki fragments. So it opens this direction. So these are all Okazaki fragments that you put them together, because this is a 5', then you make 5' to 3'. Alright? So this becomes, they call this a leading strand. Leading strand. And this is called lagging strand. Right? And there is another enzyme is called ligase. Ligase seals Okazaki fragments together. So starting with this, helicase opens it up, and one other thing, the topoisomerase works somewhere down here, because this is a twisted strand of DNA. So topoisomerase down here, once you open it up, if you the strain, that releases that strand. Topoisomerase. Okay. 3', 5', 3'. We have a primer, DNA polymerase, helicase, topoisomerase, and lagging strand, we have ligase that seals this together and creates this strand. Any question about this?
I still don't get what like Okazaki fragments are.
If this is a, let's say 5 to 3', right? So the transcription goes this direction, right? So you end up getting, so if I want to make it in this direction, you have, let's say 5', 3', right?
Mm-hmm.
But when you make the DNA, it has to be 5' to 3'. So you make this, in other words, this is done in this direction. So you open this, you make 5', 3', and then this is done. But then now it's open in this direction. So you hop back and you make this one, because as it opens it up, the direction in this strand is this way, opposite way, but you're opening the ring, the helicase this way. Okay, in the leading strand, as you open it, you start making it 5', 3' goes this direction. But when you open it this way, you make this 5', 3', but then the next part is open, so you have to go back and make 5', 3'. Since it's the reverse of opening, that's how you end up with this segment.
Okay. But what is it like? What are the fragments like? Are they
The
other
reason
enzymes
is,
or
yeah,
DNA?
the reason is because it's a 5' to 3' direction. So in this strand, the direction of replication is opposite of the way you open the ring, open the strand. So you're opening it this way, but you have to make it opposite direction. If I draw it this way, probably that makes a bit more sense. Okay, so this is a 5', this is 3'. So the direction has to be, and let's say this is a 3'. So it's 5' to 3'. As you open it up, you keep making this, and then the next part, I want you to consider it this way. So you make this, and then you open up this part, now corresponds to this part. Whatever the base pair is, you make a base pair, so it goes this direction. Now, to the lower strand from 3' to 5' strand, the direction of helicase is this way. If you want to make a new strand, it has to be 5', 3'. So this is a 5' and this is a 3'. Right at the replication fork, you are at this position, you make some nucleotides. You put them here, you make a strand. But then a second later, you notice that now this part becomes exposed. I already made this, but now this is exposed. 3', 5'. Then you have to go back and start making this direction, so you make another fragment. As it goes on, so you make another fragment here. It's a discontinuous. It doesn't follow this as you open, you read it and you make it. So you make one and then you notice backward there is another exposed part of exposed DNA. You have to go back and then go the same direction. So this causes to create these fragments. And it's also called discontinuous. Whereas this one is a continuous one because the direction of replicating DNA is the same direction as you opening the helicase. I'll try to find some pictures in the books and refer you to that if the drawing could be, it's a bit confusing. But these are a couple of things that you need to know. So this is called a leading strand, 3', 5', because the direction of synthesizing DNA replicating is 5' to 3'. Starting DNA primer, DNA polymerase helicase. The opposite direction, or the strand, the DNA original strand is 5', 3'. When you make a DNA, is the opposite direction, so it's a lagging strand, discontinuous strand, or the one that gives you Okazaki fragments. Once you make this at the end, you have to seal them together. You use ligase to seal all these Okazaki fragments together. Some of the books I've seen, they draw it this way. They just draw half of it. So they say, like this, if this is a 5', 3', 3', 5', so therefore you make this one is the leading strand, and this is the lagging strand. If that helps, half of it, some books, they write it this way. All right, so this is a duplication. Please find a section in your book, study them if you need still more clarity, and then when we see you next time, if you have a question, we'll go over them again in great details. Now let's see what is the difference between prokaryotes, eukaryotes when it comes to replication. So we're going to do a comparison between these two. Again, you're replicating DNA. The only thing I want you to be careful is primer, is the RNA primer that comes in. When I do a comparison, I tell you some of the enzymes they do. So they work to proofread. I said if there is any change in DNA sequence, it's called mutation, right? And it's very important that the cell does it perfectly. So when you add all these base pairs, there could be a case that instead of adding thymine, you add adenine. So that's the part that you need to have proofreading, or they call it exonuclease activity. And we have different DNA polymerase that they work once you make a replication, they kind of check to see if there is any base pair problem, and then they fix it. That's how the body prevents any mutation at a larger scale. Sometimes happens, passes away, that's why you have mutation, but most of the time the body is able to find that problem. So this is a prokaryotes, and this is a eukaryotes. Okay, so the origin of replication in prokaryotes, the way it works, prokaryotes has a plasmid strand. So what happens here, it creates a structure that one part starts translating, and then inside you start getting something like this, and you've heard that looks like a theta replication, because it's a single-stranded plasmid. So imagine there is a circle, you pull, you start kind of circulating inside, and then you get something inside inside the cell. Whereas in eukaryotes, they have multiple origin of replication. So we have multiple origin of replication. So DNA, so you start replicating at different regions. So you start replicating and then downstream there is another part that is non-replication. In prokaryotes, we have 3 DNA polymerase. They call it DNA polymerase 1, DNA polymerase 2, and DNA polymerase 3. Again, I hope that you're able to distinguish and separate them, that this is a replication, right? When we get to RNA polymerase, the story changes. They have different combination. So what is the function of DNA polymerase? DNA polymerase 1 goes from 5' to 3'. So polymerizes DNA, right? When you make a DNA, it has to be 5', 3'. And DNA polymerase 2, the function the book says is unknown. They don't know, they haven't come up with something to explain what DNA polymerase 2 is. And DNA polymerase 3, it has, it extends the strand, and it's fastest, complementary to this. One other thing I want you to know about DNA polymerase 1, not only it starts at least 5', 3', it has exonuclease activity. And the exonuclease activity has to be 3' to 5', or what is called proofreading. So if you make this DNA, in fact, what I have here in my note, both DNA polymerase 1 and DNA polymerase 3, they have exonuclease activity. So consider this. This is a DNA strand, right? And you're making, you're making this, right? And these are the Okazaki fragment. This is a 5', and this is a 3'. So the direction of replication is this way, right? But if as you replicate, as you go forward, what if there is a base pair here that is wrong? What if here there is something that you put, let's say thymine is supposed to be uracil, supposed to be, let's say, guanine. For these ones, what they have, they're kind of going forward, 5', 3', but they look back, they find that there is a mismatch, and they go opposite direction to fix that, and they go backward. So that every time you have exonuclease activity, the only thing in this that you have 3' to 5' direction when it comes to proofreading. So this is the direction 5', 3', this is the direction of 5', 3'. So they go this way, they read it this way, but when they want to proofread it, they make a U-turn, go back, and do a proofreading this direction. That's what's called exonuclease activity, and that's the only time you have 3' to 5' active activity when it comes to replication. But for this one, in eukaryotes, you only have one DNA polymerase. If you think of it evolutionary, so yes, in bacteria they had DNA polymerase 1 and then DNA polymerase 3. Both of them, they have exonuclease activity, but when it came to eukaryotes, they said just DNA polymerase 1 can do everything, can do proofreading, can make an extension, and everything is just done in one place. Okay. That's all I have for this part. I did talk about DNA double-stranded, no OH, and then RNA is a single-stranded and has an OH. We also did go over the mRNA type of mRNA. We've gone this, but this since I'm going to prepare you for the translation, let's just write it out so you have a million notes. When it gets to this replication, transcription, translation in gene, I tend to separate them as much as I can, but then they're so interconnected. So we were talking about DNA mutation, and you saw me talking about codons and mRNA. So I hope you are able to make that distinction. I don't want you to get confused. At the same time, you need to somehow to connect the whole picture together. So once we get to the end of translation, I pause, and then I'll ask you to help me to go over all of them one step, so it'll be more clear. Again, the types of RNA. In mRNA, if it's in a prokaryotes, it's called polycistronic. Polycistronic. What does polycistronic means? One codon responsible, one codon for multiple protein. In eukaryotes, it's monocistronic. Means one codon codes for one amino acids. I would say I'm going to fix this to amino acids, protein or amino acids. One codon codes for one amino acid. Now that I have them here, RNA is ribosomal RNA is in nucleolus, and then has codes to make ribosome. I want to add a little bit more of mRNA. I'll get to that in a moment. tRNA is in a cytoplasm, brings anticodon to small two ribosomes on ribosomal subunits. We'll go over mRNA processing in a moment, but one thing I want you to know, if this is it, again, we're done with the replication, we're about to go transcription. If this is a DNA, you make mRNA from this, you process it, process mRNA, you take the introns out, and so mRNA has exons, introns. You take the introns out, so out the introns, and whatever is left with mRNA exons. And then from here you take it outside the nucleus. So this is a nucleus, and you take it outside the nucleus to cytoplasm for translation. I'll go over the mRNA processing, that you put a 5' guanine and 3' polyadenylin. They call it triple A kel and poly G gap. So you do this to this one to make it stable, take it out for transcription or translation. Okay, so the next 2-3 topics, when it gets to transcription and translation, it's also a little bit not difficult, but lots of stuff to memorize it. Why don't we take a 5-minute break? We come back, we do transcription. Okay? What other classes are you guys taking besides bio over the winter break?
Nothing else, pretty much.
Art.
That's it. Keeps me busy. So there's no class in January, so alright. Are you guys planning to travel for the 2 days you're off Christmas?
Or just go eat somewhere, that's it.
What do usually Chinese do for Christmas?
Chinese food.
Pretty much.
You guys all from China or from Taiwan?
My parents are from Taiwan.
My parents are from China.
Mainland.
Yeah.
So is there a difference between mainland China and Taiwan?
In terms of food, kind of, I guess.
Difference in food?
Yeah, well, China, like, there's different food all over the place. Like, there's no, like, one kind of Chinese food, I guess.
Except for no noodles are.
Oh, yeah, yes. But there are differences
Yeah.
from province to province.
And how about cookies? I know there is a place on Yanzhou as you go up right across 99 branch market. It's called Kiwa or Waki. I can't remember which one comes first. It's a Chinese, very nice, this like pineapple cake, and they sell it to you.
Oh, like shunky. That one.
I
Yeah,
think that's
yeah.
also-
Yeah, that one's fun.
Do you know that?
Yeah. It's pretty good.
I went the other day, I went to 99 branch market right here on the Yanzhou, and they have pineapple cakes. That they said it's from Taiwan. They look very nice. So, something to think about. Alright, so we have a lot to cover. Let's get to the next part, which is called transcription. Transcription. Okay, transcription again, we go from DNA to RNA. And this is the part, the note that I asked you to forget about it, that comes in handy. And let's just start right off the bat. Let's say this is a DNA molecule. I want to make a few points here just for you to, when it comes to tests. If you study this well, you can easily tell me what is the RNA very quickly by looking at the DNA. You don't have to keep transcribing it. So here we go, just as a practice. Vivian, tell me what goes in here, please. A. Oh, I'm in DNA. She said U, but she thought I'm doing RNA, but this is a DNA double helix. What is a complementary? So 5 prime, 3 prime. And then one thing I told you, so which direction is transcription? 5 prime, 3 prime, or 3 prime, 5 prime? Everything is 5 prime to 3 prime except exonuclease activity, DNA polymerase 1 in eukaryotes, DNA polymerase 1 and 3 in prokaryotes. If you ever get confused, think about the evolution. The bacteria, they had 2 DNA to take care of one thing, but then as they evolved, they said just one can do it, so DNA polymerase 1. The first part of class, that replication, I didn't make it very attractive because I want to make sure to get the replication down. Now you have some idea, then we can build on that. So I'll ask more questions. So this is a 5 prime to 3 prime activity. So if that's the case, so these are the 2-stranded DNA. Which one of them? So when it comes to RNA, again, I guess I should tell you, not that very enzyme. The enzyme is called RNA polymerase, right? RNA polymerase. That polymerizes, make it RNA, and what happens that you need to you have to do 5 prime to 3 prime. So this is a 5 prime, 3 prime, and I want to see which ones do you make. So the RNA is made of, the RNA has to be 5 prime to 3 prime and is made of AUG. So if you go in this direction, right, if you start this direction, this is the strand that you have to translate. So this, you translate this, transcribe this into this becomes 5 prime, 3 prime, right? So this is, how do I say this? So this is called template, which is the antisense, and or we call it anticodon. RNA polymerase reads this to make RNA or mRNA. Alright, so 3 prime, 5 prime, 3 prime. So if you read this, what happens to this strand? This is called coding strand. And one thing I want you to know, this doesn't get transcribed. Doesn't get transcribed. Coding strand, another name for coding strand, if this is an anti-coding sense strand, this is also called sense strand. And it depends on which direction you open it. So if you start opening it this way, if you go this direction, so you're the one, you're going to get this strand, which is eventually gets 5 prime, 3 prime. Alright, so Vivian, can you help me with the mRNA for this? T becomes A, A, I'll give you a strand of DNA and I ask you what is mRNA. This is a shortcut that you can get mRNA right off the bat. So 5 prime, 3 prime, 3 prime, 5 prime, you know you have to read this because if you read this you get a 5 to 3. Now I want you to pay attention to this part. So if you have this part, what you have to do first, if you're kind of novice, you say, oh, let's try to do this. So T-A-U-G, not that she is, but I made her do this for this. But I also want you to pay attention to this part. What is the difference between this and this?
The U's or G's.
Right? So if I give you a DNA strand, if I ask you what is an RNA, in practice, yes, we say, Ali, I'm looking for the antisense template, retemplate to get this one. But if you want to be smart, find the 3 prime, 5 prime. That's your antisense template. That's fine. But then find the 5 prime, 3 prime. All you need to do, add changes T into U, and you get your mRNA. The reason I wrote this in this format is specifically because AUG, what is AUG called in terms of codon? Start codon. Start codon in prokaryotes, they call it, what is the name of amino acid? Methionine. Methionine. In eukaryotes, they call it methionine. In prokaryote, you might have heard they call it F-methionine, F-M-E-T. But either way, I'll go over this again. So methionine in eukaryotes, and when it comes in prokaryotes, it's just called F-methionine. So you get a start codon, and you go up all the way, and there are the 3 stop codon that when you get there, you have to stop. So RNA polymerase, let's just add more information about RNA polymerase. If I draw this in a form, it looks this. So this is a 5 prime, 3 prime, and this is a 5 prime, 3 prime. So this is my DNA, right? If I unheal it, RNA polymerase is here. If RNA polymerase goes this direction, which one of these is a sense? Which one of these anti-sense? Victoria. Top or bottom?
Antisense is bottom one.
Which one would RNA polymerase transcribe into mRNA?
The 3 prime to 5 prime.
This is strand. Why is that?
Because that way when it reads, it'll go to 5 prime to 3 prime and because they're anti-parallel.
Everything is 5 prime to 3 prime, so this has to be this one because this is going this direction. As it transcribes it, becomes 5 prime to 3 prime, and then it starts adding base pair coding instruction. So again, this is called template, and this is called non-coding, anti-intuitive. Welcome to biology. And you make this. In other words, this is the code that tells you what mRNA, what the name is, and non-coding, and this is the coding strand as we went over this. Okay, about RNA polymerase. So, if I add a couple of points here next to RNA polymerase, elongates 5 prime, 3 prime. Okay, can anybody complete this sentence? Reads template. Which direction does it read the template?
3 prime to 5 prime.
Very good. 3 prime to 5 prime. If you get the concept, no matter how they twist the question, you should be able to answer this. I've been telling you that everything is 3 prime, 5 prime, but then Kevin said, well, RNA polymerase read the template 3 prime, 5 prime. It makes sense. It has to be 3 prime, 5 prime because what you make is 5 to 3, and it has. This is the part that is quite important. Did DNA polymerase had any exonuclease activity? Did DNA polymerase in DNA replication had any exonuclease code proofreading activity?
Yeah.
In DNA it's very important. If you make a mistake, you want to fix it right there. That's why the cell says, I have DNA polymerase, there is a mismatch, I'll fix it, and then I'll send it. When it comes to RNA polymerase, this has no exonuclease activity. So what is the result of this if there is no exonuclease activity? All the mutation we went over, right? There is a C to G, G to A, there is a mutation. So because there is no exonuclease activity, therefore it leads to all mutation. But we do process the mRNA before it goes out of the nucleus, and it has also, the only thing I have in my note, it has its own helicase activity. You might say, Ali, when it was in a DNA replication, you told me all about the enzymes, helicase, topoisomerase, guanase, this opens up, this closes them up. No, in DNA, in RNA, it's just one RNA polymerase, does pretty much the whole thing. The trade-off is no exonuclease activity, and that leads to mutation, possible mutation. Okay. One other thing I want to mention, there is a part on DNA that on DNA that RNA polymerase binds and starts activity, and that is called promoter. So promoter is the place where RNA polymerase binds and starts transcription. I'll go in details when I talk about the bacteria for this one. Now that I mentioned promoter, you also hear about operator. I'll make it easy for you to memorize. What is promoter start with? What is the letter in promoter? What is the RNA polymerase? Has P. Polymerase binds to promoter. What is operator? If somebody operates device, operates the machine, if you want that machine to stop or do anything, you come to operator, you tell them either stop or keep working. So what binds to operator are either it's called suppressor, not oppressor, suppressor. I know one of them is inducer. What is the operator?
Repressor.
What is it?
I think it's repressor.
Repressor, okay, repressor. I do that. So either inducer and repressor, they bind to operate, inducer, repressor binds to operator, RNA polymerase binds to promoter. And in terms of location, when it comes to gene, let's hold on to that. I'll come back in a moment. So are we okay with this stuff? Again, we are in transcription. Making RNA is a fast process. There is no exonuclease activity. One enzyme does it all, that's called RNA polymerase. 5 prime, 3 prime, you know what strand, lagging strand, leading strand, and if I give you DNA, you know the shortcut, how to get to mRNA quick and easy. Okay, very good. One other thing that I have in my notice here, if this is a DNA that you make, this part, usually the part that is away from the direction of making DNA is called upstream or negative, and the one that is going toward it, this is called downstream or positive side. If you hear these terms, I want you to be familiar with them. So you start binding to promoter, start doing all stuff, and then you get at one point that is called termination. Usually there is a sequence in the DNA that signals for termination. So I don't know top of my head what is termination, but I guess it's a region that codes for the 3-stop codon. Makes sense, right? If this is my DNA, it's a region that I find that this codes for those three-stop codon, UAG, UGA, UAA. These are called stop codon. So any sequence that codes for these three causes transcription to halt and stop and terminate, right? And you make a single-stranded DNA. RNA. RNA single strand is very stable, whereas DNA is unstable in a single strand. All right, now let's talk about transcription regulation. Before I go any further, anybody has any question? So we started off with DNA. You should know which one is being translated into mRNA. You should know the start codon and the stop codon. Well, one other question I want to make. I've seen that in some of the tests. They give you a sequence of DNA, right? Hear me out for this. Let's say I have A, C, G, T, A, C, A, G, G, C, G, T. I know, okay, so this is a DNA that I have 3 prime, 5 prime DNA, and then I start translating this. So what would this be? Would be A, U, G, C, and then the rest here. A, U, G, U, C, C, G, C, A. They give you the structure. They say this is the start, we start from here. And they say, what is the sequence of polypeptide you get? Common mistake is this, oh, that's easy. I'll make mRNA. What is UGC? Let's say arginine, methionine, what have you. Yes, you transcribe this mRNA, but when you start translating, the translation has to start at the start codon. Which one is the start codon? AUG. So the peptide that you make doesn't include this because this is not a start codon. You scan this to find the start codon, and the direction is 5 prime, 3 prime. So the sequence of amino acid you make from this stranded of DNA, once you transcribe it to RNA, starts from here onward. So the first one is called methionine, and then followed by the other one. Be careful for this one. Find the start codon, from there onward start making your peptide or polypeptide. Stop codon you started, RNA polymerase that does all these activities. All right, let's go on to transcription regulation. I kind of a little bit touched on that, but let's just put it in one format compared to eukaryotes and prokaryotes. Okay. Regulation for transcription. Okay. This is, why don't I start with prokaryotes and give you an example. Prokaryotes, so this is what they have. They have, let's say if this is a gene for prokaryotes, there is P here, there is O, and this is a lag opron. For those of you who've taken, they use this gene a lot, lag opron, and then they say this is a Z gene, this is Y, and this is alpha. So these are the genes for lactose metabolism. And since yesterday, lactose is a disaccharide, is made of galactose and glucose. So this is called promoter, operon, and these are the gene. And right here they call it upstream. This upstream, and these are so what happens if this is a gene in DNA? There are some sequences upstream that tells you promoter is coming up. So RNA polymerase binds to promoter. Now, if there is no lactose, once you transcribe a gene, the process of translation, when you start making protein, is very costly for the cell, right? So the cell has to decide whether it's worth it to transcribe and then translate it, because if you translate, you make enzyme and protein. That's the whole regulatory effect of operon here is, if there is no lactose, you don't want to transcribe any of them because this break down the lactose. You don't want to make an enzyme that there is no use for it. Besides, it's a cost degradation. If there is no lactose, what happens, a repressor will come in and binds to this one. This is a repressor, and it prevents RNA polymerase to go forward. If there is a lactose, something binds to this, pulls it out, that's called inducer. You pull this out and it starts transcribing and going forward. Okay. So this is in prokaryotes. So we have lac operon, that's what it's called, lac operon for prokaryotes. No lactose, something binds. If there is a lactose, nothing binds to it. How about in eukaryotes? So I'm just going to do it here. Eukaryotes, they have, they call it RNA polymerase 2. The promoters have a sequence called Tata box. You might have heard this. So if this is the gene, if this is a promoter, they have something at upstream called TATT. It doesn't have to be TATA. It could be TATTTA, combination of thymine and adenine. So they have this Tata box of about 25 or 30 base pair upstream that kind of signal transcription. And they refer to as regulatory sequence, and about it's a long sequence, about 1,000 base pair away from promoter. So about 1,000 base pair away from promoter. And this is an operon, and these are, let's say, Z1A, if that's the gene. So that's about regulation. So you should know polymerase binds to promoter, suppressor or inducer binds to operator. Operator is after promoter, just before the genes, and depending whether you need to transcribe this, inducer operator, they want to regulate this. Are we okay for this? So these are all on DNA. This is a DNA, and you decide to comment on. Let's make a comparison between transcription in prokaryotes and eukaryotes. I'm just going to erase this. I'm writing and erasing because not as much space as you have. Okay. So again, prokaryotes and eukaryotes, and this is a transcription. So the first one, in prokaryotes, we have RNA polymerase that makes makes all RNA. In eukaryotes, and by the way, where would this happen? Happens in cytoplasm, because prokaryotes, they don't have a nucleus. In eukaryotes, we have 3 RNA polymerase. Victoria, how many DNA polymerase did we have in eukaryotes?
There's one,
How many DNA polymerase we had in prokaryotes?
3.
The second one was unknown, the first entry exonuclease activity. Okay, so that's the DNA. DNA polymerase means for replication. Okay, now let's switch to RNA. I want you to appreciate this one. So this is called RNA polymerase 1, 2, and 3. Why is it that in eukaryotes, in animals, the RNA processing or RNA is more intricate, more developed than prokaryotes? Because we have nucleus, so there are some of them they have to mate in the nucleus, for nucleus, some of the RNA that goes outside the nucleus, some of them have to make protein. Whereas in prokaryotes, they don't have nucleus, they have just one cell, one cytoplasm, so one RNA polymerase does everything. I hope you really appreciate the fact that when it gets to RNA processing or transcription, eukaryotes are more developed because we have compartments, organelle, nucleus. That's why you want to make protein. Protein makes the body. Whereas prokaryotes, just one cell, they make everything in one place. What would RNA polymerase 1 make? And again, you have how many type of RNA? 3. So they have to make, something has to make them. So RNA polymerase 1 makes rRNA. RNA polymerase 2 makes mRNA. And RNA polymerase 3 makes the tRNA. The way you can memorize it is 3 starts with 3, RNA polymerase 3 has T at the end, at the beginning, tRNA has T. And if you remember 1, 2, 3, RMT, however it is, every time it comes to RNA, I like to start with rRNA, mRNA, tRNA, because this kind of stays in students' mind, and then when I say what makes them, 1, 2, 3 goes in sequence. You need to know this. So eukaryotes, you have these 3 mRNA. For prokaryotes, transcription, and translation happens at the same time. If there is no nucleus, it's everything in the cytoplasm, so there you don't want to separate them, or you can't, there is no way for you to separate them because all in one place. So at the same time, and there is no time for modification. Okay. This one, so this one, there is a modification, and I kind of went over this slightly, and that is called mRNA processing. Vivian, what do you make in mRNA processing? What comes out and what goes in? Exon, intron, exon. 5 prime to 3 prime. So if I do processing, what is it that I take out?
Intron.
The way I memorize it is extinct but expressed. Exons are being expressed, so you have to keep them, you express them, you transcribe them. Introns, intruders, you take them out, you push them away. So this would be, let's say, exon 1, exon 2 would be exon 1 and exon 2. What else do you do when you process this in addition to take exons out? You add 5 prime, some of the books they say 5 prime cap. but I want you to know a little bit more. You add 5' cap, but what kind of amino acid do you add to the 5' specifically? Anybody? Guanine. You add 5'. They say cap, but please know 5' guanine cap, and the 3' they call it poly A tail. You add A. 5' cap and poly A tail. So sometimes this process is called, I keep referring to our processing, sometimes they call it splicing. Splicing, as Vivian said, means taking the introns out and keeping the exons. 5' cap adds 5' to the end. What is the purpose of this? This enables ribosomes to find mRNA. So enables ribosomes to recognize 5' part of mRNA. I didn't know about this until I saw this. I somehow forget this. The poly A tail in my notes that I have, it says helps you in, let's say, helps you determine half-life of mRNA. I guess there is a mRNA degradation, there is a half-life, so the amount of A helps you find out what is the half-life of mRNA. All right. Anything else that anybody wants to ask or add, please let me know. This is pretty much all about transcription. This happens in the nucleus. You make mRNA, you take it out. Well, in prokaryotes, we have only one polymer, one RNA polymerase. In eukaryotes, you have 3, but the one that works in transcription is only RNA polymerase 2. In a sense, in both eukaryotes and prokaryotes, there is one RNA polymerase that does transcription. For prokaryotes, that's just referred RNA polymerase. For eukaryotes, we call it RNA polymerase 2. If you ever get confused, think about in eukaryotes, in human, there is more investment and processing done from to make mRNA into protein because our cells is compartmentalized, means we have nucleus and organelle, so you have to have 3 different RNA polymerase for each. Going backward, if it's a DNA duplication, in human we are more developed, so you make only one DNA polymerase that does everything, whereas prokaryotes there are 2. Alright, let's get ready for translation. So we started with DNA, sometimes we duplicate it, and then we decided to transcribe it. Now that we transcribe this, we have to start making protein. That's the investment. That's the harvesting part. Everything we did just to make this. And this is the part that is very costly for the cell. Highly energetic. You have to use energy, but the positive is that you make protein, enzymes are protein, what have you. All right. So translation. Vivian, where did transcription start? Nucleus. And what did you make as a result of transcription? Where does mRNA where is it being made? Nucleus. But then you process it. Where do you process mRNA? In the nucleus or in cytoplasm? In nucleus. Everything you do in the nucleus. Once you kind of prep and spruce up mRNA, everything's done, you send it out into the cytoplasm. It comes through a nuclear pore, it comes out. Now nuclear mRNA, process mRNA, no intron, 5' G cap, poly A cap, nice and easy, comes to cytoplasm to be translated. So translation happens in cytoplasm. Okay. Again, again, prokaryotes ribosomes. I'll go over this when I talk about prokaryotes, eukaryotes. What is organelle? Can anybody describe organelle?
A small specialized part of the cell.
Okay. Very good. So there are, let's say, he was trying to be as eloquent as he could, but that's the best he could do. Neither I can do anything else. Small specialized part of the cell. Can you be specific about the membrane? How many membrane would an organelle have? 1 or 2?
Oh, some have one and some have two.
Is nucleus an organelle?
Yeah.
Is mitochondria an organelle?
Yeah.
Let's switch to plant. Is chloroplast an organelle?
Yeah.
What do they all have in common? They have 2 membranes. Okay, hold that thought. Is ribosome an organelle? Okay, what is the difference? Prokaryotes, eukaryotes. We say prokaryotes, they don't have organelle. Eukaryotes, they do have organelle. Right? Okay, why is this that I say prokaryotes ribosome and eukaryotes ribosome? I thought ribosome. I thought prokaryotes, they don't have an organelle. So first, ribosome is not an organelle because you see it in prokaryotes, eukaryotes. So if it's not an organelle, ribosomes must be single layer. Anything that is a single layer is not an organelle. Everything in our cell, they are double layer, they are organelle. The only thing that is single layer, therefore, is not an organelle, is ribosome. That's why we see ribosome in both prokaryotes and eukaryotes. In prokaryotes, they call it the 70S, which is made of two ribosomes, 2 subunits. One is called 30S plus 50S, and in eukaryotes, they call 80S, which is made of 48S plus 60S. What does S stand for here? Different books say different things. They say the first letter of a Swedish scientist, and some people they say it's a sedimentation rate. So I like sedimentation rate. There are bigger subunits, so the sedimentation rate, the sediment is smaller. So 40 is 63. This is the one that they don't match to what they say. So ribosome. So are ribosome organelle, Kevin?
No.
Why not?
Because they have single membranes.
So what classifies organelle?
Whether or not they have
In
double
addition to
membranes.
small specialized, now you know if they ask you what is organelle, to be very concise and scientific, organelle is small specialized part of the cell that has 2 membranes, must have 2 membranes. Otherwise, it's not an organelle. What is the concept you can use to prove your point? Ribosomes found in both prokaryotes and eukaryotes, they cannot be organelle, so they must be single membrane, which they are. All right, it comes in here, and then so this was where you make peptide bond in cytoplasm, and let's just start with this. I wish I had a picture in the book. I'll try to here. Let's see. If you give me for one moment, I will be able to find a good picture. I don't mind drawing it, but sometimes the book is wonderful. Okay, here we go. Oh, I'm about to get knocked out. Here we go. 3rd time is the charm, right? Please pick it up. Okay, so let's go here. And then I want to say translation. And I may get some good images. Let's start with this one. Sounds. Okay, so very briefly we went over this transcription. They're Deutsch, they're German, so that's why they put K here. Transcription, we go DNA to mRNA. mRNA comes up as a mixed polypeptide, pre-mRNA, that's perfectly good. So this is the part that you I'll go over this and then I'll put the details on the board. So this is the mRNA that comes in. First it comes to the nucleus, to cytoplasm. Small subunits comes in, lays on small subunits. And big subunit, when it comes and binds, there are three sites. One is called A site, P site, E site. Think about A as approaching, P just a placing, and E as an exit. So the very first amino acid that comes in, it comes into a P site because A site is already empty. We don't have anything to make of. First amino acid goes to A site, and then the next amino acid would come to A site, and then you have to make a bond between them. I emphasize making a bond because I want to tell you how many ATP you use, how much energy you need to make this one. So the first goes to A site, the next one comes to P site, the next goes to the A site. You make a bond between them, polypeptide bond, and then once this is done, there is a translocation. You put everything into E site. So these two, whatever is here, they translocate into this. A site becomes empty. The next amino acid comes in, binds with this one, and then it goes to the next site, and the one on the E side being transcribed. So it's the first protein, first amino acid, and that's how you do. So if this is mRNA, it kind of goes through this. This stays, ribosome stays stationary. This gets kind of like a thread, goes through this, and then for each 3 base pair, you make a peptide, and it comes out, and that's how you make the polypeptide chain, right? Small, big, APE, first amino acid goes to the P, next comes here, and then you make a bond translocation to go forward. I wonder if they have a DNA structure. So let's see if I have another one. Let's do something to close this. That sounds. This is a very repeatable journal, PNAS. This is a high in the journal, and that would be very descriptive. They just talk about the factors. But you got the idea, right? So how this one comes in, and then let's see one more, and then we move on. That sounds interesting. EDU blog. Everybody have a blog these days. That's another way to think about this. So, where is the first one, I guess? The first one probably, they didn't say the first one. They did opposite way, so this is the E side. But you got the point. So that's the whole notion of getting, finding DNAs. Alright, so let's go over the details. So you got the picture of how it works. This is my mRNA. This is again 5' to 3', comes in the nucleus. We have a small subunit, and then once it stabilizes, the big subunit comes in here. We have A site, we have P site. Some books they just say A and P, and then this pops out, but we can say E site. The first amino acid will go into here, and by the way, what brings that amino acid here? tRNA. So let's say this is a AUG start codon. tRNA goes into nucleotide, into cytoplasm, binds this one. So AUG, the amino acid, the structure of tRNA look like this. This is how. Prime, 2, 3 prime. Okay, so this is called, I doubt that you need to know for this is called D loop, and this is an anti-codon loop, and this is called variable loop, something here, variable loop, 5 prime, 3 prime, that's the structure of amino acid. So whatever this side is, this is the part that matches the variable. I guess one side is an anti-code loop. So this is the part that I guess matches with the amino acid. Let's say if this is an amino acid that I want to bring here, the codon is AUG, so this amino acid has to have, so what would the, oh no, so this is how it works. This recognizes AUG, the anticodon comes in, picks up a methionine amino acid, attaches into this and brings it and just place it on top of this. But the anticodon for AUG problem is UAC. So UAC amino acid, this matches with the UAC, picks it up, brings and places it into this. That's the first amino acid comes in, and then the next one, let's say CGC, CGC comes here. Then between these two, you have to make a bond. To make a bond between these two, you translocate this into this. So then you get AUG, CGC, and this is the A side vacant, and the next one comes in, and eventually this becomes, you start threading this. Let's say the next one, CGC and AUG amino acid. That's how you make these structures. Very straightforward. The next part. Okay. I wanna in the book it is I have it very extensive, like writing how many energy you want or how this transformation translocation takes place. One thing I want you to know is this: the process. So it starts three process in eukaryotes. First one is called initiation. Initiation means initiation is, these are the events happens. 30 as again, this is in prokaryotes. 38 bonds to a P site. Okay. In number 2, F-methionine. If it's a prokaryote, F-methionine, tRNA brings methionine amino acid, methionine amino acids. So number 2 is called elongation. For elongation, I will write the energy. Initiation requires 1 GTP. Elongation requires 1 GTP. So elongation not only is a peptide, is a transferase, so it's a peptide transferase. It also requires translocation. Goes from A site to P site. The last part is called termination. And this is stop codon, where the stop codon comes in A site. So when you have stop codon at the A site, this has to stop. Okay. Eukaryotes transcription is pretty much the same as this, but instead of F-methionine, you only have methionine. Alright, the last part that I want to talk about, I did emphasize that the process of translation requires energy. You have to use lots of energy. Let's say if you want to make a 50 polypeptide chain, how much energy you need, and I'll give you a shortcut to find the amount of energy. So I'm going to erase this, and we'll talk about the energy and formula to find out the number of GTP or ATP that you need to have to make a polypeptide chain. So for this part, so how much energy we need? The first part, let's say this is about energy for translation. The first part is called activation of, let's say, activation of, let's say, tRNA with amino acid. So, for instance, if you want to make a 50 amino acid polypeptide chain, you want to have 50 amino acid. For this one, you need 2 ATP, let's say 2 GTP per activation. So it's 2 times 50 becomes 100. The second part is called elongation, and I like to say translocation. How many times, if you want to make a 50 amino acid, how many translocation or elongation do you need? For how many amino acids? There is a caveat, there is a catch in this. Is this 50 or anything other than that? Remember, the first one goes to the P side, right? So the first one is empty, already there. Anything else, you have to bring it and then move everything upward. For elongation, this is a 49 times because the first one, the first amino acid. Some of you might say this, some of you might say, Ali, this is an A, this is a P, this is an E side. In some books they only use A and P, but the point is when it goes to the exit side, this is the part that you start making the branch. So elongation or translocation is anything that you hop over from A to P side. This is what I mean by elongation, translocation. From here to exit, you don't use energy. So for elongation part, since the first one comes here, you need to do it 49 times to make it polypeptide chains. That's what it's because the first amino acid is placed in P side. P side, no translocation required. And then the third one, it's called initiation. For this one, so if it's a 49 and you need 1 GTP, right? Or for this one, you also need 2 GTP, 49, and then 49 times 2 GTP per location, per elongation. 49 times 2 is 98. And then initiation, just to bind tRNA to amino acid requires 1. No, that's about elongation. Initiation, to start with, just to assemble this, for the first one to bind to P side, you need 1 GTP. Okay, so if you add them up together, what do we get? 199. Alright, now I want you, so the reason I wrote this is if they ask you in each step how much you need, you should know that. Initiation 1, the first one. Elongation, however number of amino acids is -1. Activation, you need 2 GTP. By activation is tRNA binding to this. I want you to memorize this formula, 4n minus 1. N is the number of amino acids that you need. So if it's a 50 amino acid, 4 times 50, 200 minus 1 is 199 amino acids. Okay? Now, this is lots of energy, lots of ATP. One glucose gives you 36, right, after all the stuff we went through this. So if you want to make a 50 polypeptide chain, this is lots of energy used. Some of them, they are much longer, insulin or other enzymes, and there are so many enzymes in our body. So the process of translation is very costly, so you have to commit yourself with the cell, convince yourself whether it wants to go through this or not. That's why in the gene or DNA we have all those promoter, inducer, or repressor to decide whether you want to transcribe it or not. Alright, let's go to the last part. Telomerase, very crucial. So about telomerase, we'll take like 2-3 minutes break, and then we'll come back and we switch to the whole new story. We don't talk about all of this nonsense anymore, so we switch to viruses, and I really want to get through this. Hopefully we will. Okay, what is telomere and what is telomerase?
It's like the end of the chromosome.
They
Okay.
maintain the length of the DNA so it isn't shortened every time it goes through replication.
The one I was looking at, the end, replication. So sequence of base, nitrogen base at the end of DNA. Again, this is a telomere, and for every replication and aging, DNA gets shorter. So there is an enzyme in our body that makes the telomerase, called telomerase, that adds a nucleotide, A, to the end of DNA, and prevents shortening. Or I don't want to say destruction, shortening and the redivision. So I see whether I can explain this. So this is a 3 prime and this is a 5 prime, and this is a, okay, so this is 3 prime, 5 prime, and what you make, again, this has to do with the Kawasaki fragments at the end of DNA. So 3 prime, 5 prime, this becomes 3 prime, 5 prime, and so you have to make it this way. Adds DNA at the 3 prime, so this is the part has telomeres, and if this is 3 prime, 5 prime, this is the direction you make DNA, right? You make replication. So every time you make a replication, I guess, something happens at this end. For each age, you start, I guess, starting from here going onward, and this part is being exposed and eventually gets degraded. What the body does, it starts to making A, so whatever is A, you make T here to protect this from being exposed and being degraded. So after this division, this long chain becomes like 3 prime, 5 prime when you make this, so it becomes shorter and shorter. But the telomerase binds T into at the end of this DNA to prevent this from shortening, but this is an enzyme, it doesn't work properly at old age, therefore DNA starts getting shorter. I'll try to send you like a better picture description. Just wanted to let you know the concept of telomerase is just adds A to the 3 prime end of DNA, prevents it from shortening, and DNA degradation or shortening is a process happens at every replication and through the aging process. Okay, so that's about the DNA, about the telomeres. All right, we are pretty much done with. I don't want to say the gene. We'll come back to. I didn't write it here. It's about Mendel's law, but that's about inheritance. But in terms of structure, going to DNA, RNA, this is pretty much you need to know. make a good distinction between the replication, transcription, and translation. Now that we wrap it up, if you give me 5 minutes, one of you start telling me about what is replication, whatever you know. Who wants to go first? Vivian? Replication. What do I do in replication? Okay, so she got the difficult one because there are lots of enzymes going on. So just everything you know, tell me. Replication. You go from DNA to a DNA. Okay, semi-conservative dispersion or conservative?
Semi-conservative.
Semi-conservative. Is DNA parallel or anti-parallel? DNA is anti-parallel, right? Anti-parallel goes from 5' to 3', 3' goes to 5'. When you replicate DNA, what are the enzymes that you use?
DNA polymerase.
There you go, you said it all. DNA polymerase. In eukaryotes, how many do you have? 1, 2, or 3? In eukaryotes, we have 1. In prokaryotes, we have 3. What is the function of DNA polymerase 1 and 3 in prokaryotes?
They crack the states.
That's one DNA exonuclease activity of proofreading. Also, if it's a DNA polymerase, it polymerizes DNA. Very good. So if it polymerizes DNA, it's 5' to 3'. If it's the exonuclease activity, is a 3' to 5', right? So that's one. Another enzyme. We are in 5-minute stretch. What was it? Primase. Very good. Or, as she wants to be specific, RNA primase. What do RNA primase do? When you open the DNA, RNA primase is where you add the very first base pair to begin with. So to begin with, you add RNA primase. Then DNA polymerase at the end comes over, replaces RNA primase. Says, you know, thanks, you help us to do this, but we're done, we can take care of it. So pushes primase out and adds more. One other thing, this is the RNA primase. Just be careful, you can't add any uracil. Whatever it adds has to be ATCG. RNA primase. What opens the DNA strand? Helicase. Okay, and the place this opens the DNA strand is called replication fork. Very good. Number 4. Okay, when you open it up, if it's a twisted strand, when you open it up downstream, there could be some twisting. What unwinds, what loosens that string? Topoisomerase. Okay, what is the antagonistic of topoisomerase? What pulls them together? Guidase. If you make, so in DNA, when you make this, you make 2 different strands. One is called leading strand, one is called lagging strand. What do you have in lagging strand? There are fragments, is a Japanese name. Okazaki fragments. Okazaki fragments. What attaches all the Okazaki fragments together? Ligase. This is a bit tricky. Ligase. Ligase attaches Okazaki fragments. Okay? Very good. Happens 5', 3'. Anybody else wants to add anything to replication? Kevin and Victoria, what else did you want to add? Okay. Anything else?
That's all they have.
You could add in telomeres because they are involved in replication.
Very good. Telomerase. Vivian, what is telomerase? Enzymes that prevents shortening of DNA, adds a polytails. So this is an enzyme that adds A to prevents shortening of DNA. All right. Anything else? So that was for Vivian, the one and only Vivian. Kevin, you're next. Talk to me about transcription.
It's 5' to 3'.
Very good. DNA to RNA. Again, 5' to 3'.
And in DNA there are in eukaryotes, sorry, there are 3 RNA polymerases. And in prokaryotes there's 1. And
Yeah, pause for a moment. Vivian, you did say how many she did say 3, right?
yeah,
DNA polymerase 1, and she said, okay, very good. Prokaryotes. This is a RNA polymerase, right?
yeah.
What does it make? How many types of RNA we have?
3. So tRNA. No, wait. RNA, RMT, RNA, and RNA.
Somebody taught me this mnemonic is kind of crazy, and I doubt that you might laugh at me, but I was teaching a course at Princeton Review in Los Angeles. Someone says, hey Ali, there is a mnemonic. He says, so what is it? It says 1, 2, 3, you are empty. Whether it makes sense or not, I have no idea. 1, 2, 3, you are empty. I kind of laugh and yeah, Ali, we're using this. This works. Whatever it works. If you want to use that 1, 2, 3, you are empty nonsense, do it. All right. RMT, RNA, okay. Prokaryotes, they only have 1. Okay.
The eukaryote.
Just talk to me a little bit more about this. I want to make sure you got the concept. Why this is so sophisticated. This is only has 1, does everything.
Well, eukaryotes have nucleus, and so.
Where is mRNA is made?
It's made in the nucleus.
Nucleolus. So this has a code to make. You have to make ribosomes, right? Nothing happens de novo. The thing that where first you make ribosome comes from nucleus. Our mRNA has a code. So it's a nucleus. mRNA is made in
Nucleolus.
mRNA made in a nucleus. You should know this. This is cell, right? This is nucleus. Right inside this is a nucleolus. So mRNA is made in the nucleus. rRNA is made just like a headquarters, the very center of everything in here, and then comes to make. But this is RNA. Nucleus, nucleolus, nucleus, and then will be shipped process to cytoplasm. T.
Cytoplasm.
Cytoplasm. Prokaryotes have 1. The reason they have 3 of them because they have nucleus, they have organelle, each of them made as a specific region, and that's one of the things should give you a hint that there is some modification. Things doesn't happen at the same time. You make DNA, mRNA, you process it, and then you go to cytoplasm, get 1, 2, 3. Whereas in prokaryotes, since it's only one compartment, transcription, translation, replication happens at the same time. It's just chaos. All right. We got about this. What else you can say about transcription?
You have to have the start codon first.
What is the start codon?
That's AUG. And then the amino acids, methionine.
Methionine in eukaryotes. What is in prokaryotes? F-methionine. Very good. Stop codon. 3 of them.
Yeah. UAG, UAA, and UGA.
Anybody knows mnemonic?
You are gone, you're away, and I don't know that
You
one.
go away.
Okay.
I heard bad things. You are. No, that doesn't. Somebody say you're ugly. You something, but ugly starts here. Ugly, you go. However it is, but you are away. UAA, UGA. Crazy. I know. So these are the stop codon. Anything else? Everybody else, if you want to add anything else, please chip in. We need more of this. Come on, I spent like an hour to do this, not for this. What are we talking about?
It's based off of the template from the DNA.
Perfect. And now I'm gonna corner you. This thing, you can't get away from this. You must know this perfect.
It's the non-coding
Okay. Top or bottom?
bottom.
Bottom, because it's 5', 3', and RNA polymerase, if it goes this direction, you make the bottom. Now, on hindsight, I have another idea. What if RNA polymerase opens it this way, right? It all depends on which direction it starts. If it goes this direction, then it would be 5' to 3'. You have to read it carefully to see what it is. So, the one that you're going to transcribe to make RNA is this one. What is the name for this? 3 names. Template.
Oh, antisense,
And
anti-codon.
non-codon.
Oh, no.
Template, antisense, and non-codon. So this must be coding. Not codon. Coding, non-coding. Coding.
Sense.
Very good. You make it off of this, and then the shortcut. If I give you DNA, if I ask you want to find the structure of RNA, so 5', 3', 5', so make it. If it's going this direction, you make 5', 3'. All you need take the coding, switch whatever is G, whatever is T to U, and then that's repeat. All right. So that was it. So it's very important. So this is a template. All right. Anything else? Can you make a distinction? What is the difference between prokaryotes, eukaryotes? I talked about something called monocistronic, polycistronic. Which one? What is monocistronic, polycistronic? Remember, eukaryotes are very sophisticated, so you want to take advantage. So they must be polycistronic. Oh no, prokaryotes. Prokaryotes. Take that. I missed that again. And Vivian is laughing at us. Come on, Vivian, give us a break, would you? Okay, where is polycistronic?
Should be like,
Okay, types of RNA, mRNA, prokaryotes is polycistronic, eukaryotes monocistronic, right? So you make mRNA here, mRNA, if this is a prokaryotes and this is a eukaryotes. So if it's a prokaryotes is, can we use something to make it easy? Here we go, I got it. P for prokaryotes, poly for polycistronic. Finally, something works. Polycistronic, right? And U is monocistronic. Monocistronic. What does polycistronic mean? One codon
for multiple proteins.
multiple amino acid, right? Again, it's just not sophisticated. You don't, everything happens together. One can make different things. No idea how this works. Monocistronic, one codon is one amino acid. Was there anything else? We have in eukaryotes we have multiple origin of replication, right? Is this multiple origin of replication or multiple origin of? No, that's DNA. That's DNA. Don't mix it up. All right. Never mind. All right. So now that I found this, what Vivian said, in replication, you have multiple origin of replication. In eukaryotes, this is eukaryotes, but in prokaryotes, the DNA in prokaryotes is plasmid, like this is a cell, this is plasmid, and all it does, it kind of peels off inside. So what you make, you kind of peel it off and you make this inside the cell. That's called only one origin of replication. And even if this, it's not here, but they said it looks like a theta, means this is a structure, you start peeling it off and then make one inside. How we call it. That's a replication. All right. Anything else you want to help Kevin, everybody? We did this poly, mono, what have you. All right. Are we good? Let's go to Victoria. Victoria, talk to me about translation.
It occurs in the cytoplasm and the ribosome, and there are 3 sites: the A for approaching, P for placement, and E for exit. And we start with the AUG codon. The tRNA is like in the shape of a cross, and the
Cloverleaf.
and the bottom is the anticodon loop, and tRNA will bring AUG and methionine into the P site to start off translation.
How much energy you need?
2 GTP. And then
Was the initiation 2 GTP?
initiation 1 GTP. 1 GTP.
One just to put in a P site. Okay?
And then elongation takes 2 GTP.
How many elongates if you have 100 amino acid?
It takes 99 times 2.
99 elongation, each elongation 2 GTP, 99 times 2, 291.
198.
Okay.
And so it will, you put in like the tRNA will bring in the anticodon with the corresponding amino acid, and then
Let's say alanine.
it'll bring in, let's say, alanine, and it'll attach to the A site and then bind to the methionine, and then everything will move over one.
Okay.
And
Very
the
good.
methionine will exit, and the A site will be empty.
Okay. And then N.
And then the translocation will take 2 GTP.
Okay, we did so. Elongation or trans. Okay. I want to make it clear. Initiation 1, elongation. I guess when I say elongation takes 2, activation is the what is activation? Is the process of tRNA finding corresponding amino acid, right? This involves binding, they call it amino acyl ATP. It uses the ATP to make a bond between amino group and acyl group of tRNA. That's a costly one. The fact that tRNA has to go in the nucleus, find amino acid, binds it, stabilize it, bring it. That's what's called activation, and activation requires energy. So 2 GTP. How many do you activate?
If it's like the 100 amino acid change, it'll take 200 GTP.
Makes sense, right? For every amino acid, if I want to bind amino acid ATPase, I have to bind, secure it, bring it. So 2. This is 100 times 2, which is 200. Okay. And then what is the termination? How would terminate?
I think it'll terminate when it hits the stop codon.
What is the stop codon?
UAG,
One of these three. Stop.
UAG, UAA, UGA.
Where is the stop codon should be to terminate? When the stop codon gets to which side?
A side?
If the process keep going, comes to P, the next comes to A. Whatever it comes, approaches A, arrives A, however you call it. So as soon as the stop codon here, that stops. So it has to be A side. So initiation, you go to P side. Elongation, you just go from A to P, you hop over, you translocate. Stop has to be. So if it's if it's 100, how much the total is? Either you can do the math here.
4 and minus 1, so 399.
390. All right, it's 1125. Okay. Would that be okay? Would that be okay if I start the cell division, because this is kind of related to this, and then the next time you're going to go nervous system, and then prokaryotes, eukaryotes, I don't want to push this. This is important stuff. I can push this in like 15 minutes. This probably 10 minutes or so. Why don't we do this? We take like a 2-3 minutes break, 2-3-minute break, come back, we do cell division, and then tomorrow when we start off, I'll make sure to go over this and then we go nervous system. Right? I'm kind of raising the bar a little bit. I add more. Even yesterday, the email I sent you here, I had Mendel's law and non-segregation and genetic inheritance. That takes a little bit time, and I've seen questions you have to know with a pedigree. But for now, let's do cell division next. Tomorrow we'll do this and nervous system. Now if I do this and nervous system, then in mental inheritance, we'll figure it out. I'll try to do this by tomorrow. All right, so like 1 or 2 minute break, you come back and we'll do the rest. And I will send you test. Please make sure to take the test.
So when are we like around ending class today? In this 12 or
Oh, 12.
after 12? Okay.
The way it goes, I see. So Monday you guys could come at 8:30, right?
Yeah. Wait, when were we supposed to come this morning?
Yesterday we said Monday is at 8:30, and then I told them today I'll be here at 8:30. I came at 8. Push Sherry. I emailed Sherry. Sherry, can you come at 8? And it's like 12 midnight, and she says, Ali, why this early? I said, I think they're coming at 8:30. I want to make sure to set this up. I want to make sure everything is right. Take care of some stuff. But to her credit, she came in. Today we're supposed to be here. I told them I'll be here at 8:30, but it's alright if you guys came in with it. But from now on, let's make it 8:30. Hopefully the next students they can catch up. And then the goal is to finish by 12. But it's a law that has been broken over and over again because Ali is insufficient, doesn't work properly, doesn't keep track of time, and is always behind. Okay, forget this. I'll be right back.
You go to school in San Jose, right?
Denver.
Do you know anyone from Leland?
I know people who used to go to Leland, and I know I think I know 2 people from Leland actually, because I volunteered with them.
Do you know a sophomore named Kyle? He's like
No.
tall.
I think both of the ones I know are seniors.
Did you know Annie Mae? She graduated from Lynbrook last year.
I don't think I do.
I think you're a freshman and she was a senior.
Oh. Yeah, I knew 2 seniors when I was a freshman. Why not? Well, you go to American High, right?
Yeah, it's really far away.
Yeah, I think I know you.
It's not like a very motivated school.
Oh. Are there a lot of Asians or no?
No,
Oh.
it's not.
Opposite of them.
Yeah, I take more AP classes than some of the juniors there.
Oh. You're a sophomore?
Sophomore.
Oh, okay. We don't even have AP bio for sophomores.
Okay.
Oh, we're not supposed to, but one of my teachers,
Oh.
yeah. I had a really bad 9th grade biology teacher. And he would just give us good grades for no reason.
Oh. Well.
I can do one other thing, folks. 1, 2, 3, 4, 5, 6. There are 6 pages that I prepared for virus, prokaryotes, eukaryotes. What I can do, I can scan them and email them to you. Please study them. I like to be optimistic, but the way I see it, the way I have planned, I doubt that we can cover these mentals and go to nervous system. Why don't we do this? You take a rain check on this from me, and you study them, you have everything, and then later down the road, if you get time, I promise, come back and go over it. Alright? Deal? Deal, Vivian?
Yeah.
Victoria?
Yeah.
Okay, Kevin?
Yeah.
Alright, so this cell, let's go over. Holy cow, no, 6, 7, 8, 9, 10, 11, 12, 13, 14 pages. That's crazy. Alright, I'll get you some, but you have the rain check. We'll go over this. Cell cycle. So it consists of different phases. One is called G1, one is called S, the other one is G2. And this is called cell division, and also refers to as M phase, mitosis or meiosis phase. this part from G1, guess I should see if I can do this. From G1 all the way to G2, what is the name of this phase? Interface. What happens at G1? There is growth, but no increase in size. No increase in size. S, anybody can guess?
Replicates DNA.
Very good, replicates DNA, and to make them
More organelles.
simpler, S stands for synthesis, in the parentheses, replicates DNA. So all the replication we just learned, DNA polymerase coming, going, helicates, what have you, happens in S phase. And if it's a G2 phase, this is a further growth, but this is the part that cell makes organelle. Now, like we said we talk about it making a translation, that's a costly step. The cell needs to decide or not. Or even about glycolysis, remember PFK, phosphofructokinase was a committed step. Once you go through that hump, everything goes down the hill. The same is here. There is a step in here that once the cell passes through that, it's just you commit to finish the whole cycle. You come in, you think, you pause, you gather your thought, and then you commit, and then you go through that. That place comes at this region, and there is a gene is called P53. If you ever read any article, any news about cancer genes, they say there is a mutation in P53 gene. This is a gatekeeper. This is like a committed gatekeeper. So the committed stage or step. Once you pass through this, the cell goes through the cell cycle and finishes the mitosis. Okay. G phase, very good. Humans, how many sets of chromosomes we have?
23.
Humans, we have 23 sets of chromosomes. Okay, from 1 to 22, they call them autosomal, and 23 is called X chromosome or sex chromosome. What is karyotyping? Karyotyping. Karyotyping is a scientific method that allows you to see, to see or photograph chromosomes. Then this is the part you find the question whether there is a non-disjunction, disjunction, or missing chromosome. What happens to the size of chromosome? Chromosome 1 to chromosome, let's say 22. Which one is bigger, which one is smaller?
One is bigger.
One is bigger, as you go down, becomes smaller. I hope I can remember this right. Chromosomes also have two regions, so they call it P and Q region. I think from here to here, they divided. This is called P region, and this is called Q region. They just divided. And sometimes they say the gene for color blindness is on chromosome 11, region P, and then they have some base pair. So if you hear P or Q when they talk about a chromosome, just know they're referring to different regions in chromosome that you have that gene. Okay, couple of terminology before we delve into this. What is allele? Allele, again, this is like I'm going down here, but that's fine. What is allele?
It's a version of a gene.
Okay, let's say alleles. So alleles are two different versions, two different or identical version of gene. What is trait?
It's
characteristic.
displayed by a gene.
I wonder whether I can say this like a phenotype or characteristics. That's what it's called, characteristics. Each trait, okay, phenotype, and I want to say each trait is represented by how many alleles? 2 alleles. I say not 100%. There are some traits, some research they've done, that there's something that they can refer to more than 2 alleles, but that's not for this scope. And let's say, I don't want to, I have a feeling I'm getting away from cell division. It doesn't have to do this, but these are the things you need to know. But that's fine. Probably we go a bit later. Loci. What is loci or locus? Locus singular.
A point on the chromosome?
So, say?
It's like, I think it's like the location of the wheel on the chromosome.
Location of allele or gene on chromosome. So when I say P, Q, what have you, that's how they come up with loci, location of the chromosome alleles. Now that we talked about phenotype, phenotype, here, if I write it here, phenotype is appearance, genotype, what is it?
The actual alleles?
Genetic makeup or alleles. Alleles can be in 2 forms. Could be homozygous or heterozygous. If it's homozygous, let's say T for tall and T for short, right? So for homozygous will be TT or TT, heterozygous will be TT. I don't go further into this because I know if I do this then I'll get into Mendel's law of genetic traits. We'll come back to that, but for now these are a couple of feedbacks you need to know about gene. Let's go back to the cell cycle. Are you okay for this? All right. There are 2 different versions of cell division. One is called mitosis, the other one is called meiosis. So I'm just going to write it here, mitosis, and you guys tell me what you know. Okay, what do you know about mitosis?
4 phases.
4 phases.
You end up with a daughter cell identical to the parent.
Identical daughter cells. So could I say there is no reduction in genetic or chromosome number? So it starts with a 2N, you end with 2 and 2N, right? Identical cells, but how many identical? 2. Okay, what else? It's an autosomal or autosomal cells happen in autosomes. Every place in our body is mitosis except 2 regions, ovaries and testes. Ovaries for female, male. So in ovaries and male, you end up ovaries make egg, testes make sperm. So these are N and N. So these are called sexual division. This is called asexual division. Okay. Autosomal. Do nervous system tissue, do nervous cells divide? Okay. So everything divides except the nervous system cells. They don't divide, right? So asexual division, autosomal cells. Okay. Now I'm going to start drawing the pictures, but then let's see if you want to add anything else. How many stages are in mitosis?
4 stages.
4, some people they say 5, but we'll see it. So it's called prophase.
Metaphase.
Very good. The reason I'm hesitating to put that right in here because some of the test questions I've given you, they add in this stage.
Prometa.
You say?
Prometa.
Prometaphase. I'll write it in green, in red, because these are the ones that sometimes in the book usually they don't write it. Some teachers do some prophase, followed by metaphase. why don't you do this? This is important. I'm going to erase this, write them in here, and show you a picture of this that we can put them all together. Would that be okay? Are you okay with this, my process? And the only way you can do this is just solving, taking test problems and seeing how they are testing you. Otherwise, the concept is pretty straightforward. I mean, a few things you remember, and you. It is an art how to use these erasers and be productive. So at some point, I find myself keep capping them instead of writing. That's crazy. I wish I could write with both hands. All right, here we go. Let's see if I can do this. No, it doesn't work. All right, prophase. Again, we are in mitosis at 4th stage because it has only one prophase and the rest. What happens in prophase?
Chromosomes condense.
Condensation or condense chromosome. Condensation is different. Condense chromosome. Okay. Okay, what else? These are easy ones. No offense, easy ones, but I want to ask you a question again. What else? What happens to nuclear membrane?
Oh, it dissolves.
So nuclear membrane starts to break down, dissolves. Okay. When chromosomes condense, this is the part that chromatin, these meshes that chromatin becomes chromosomes. And you start seeing what? You start seeing this is a chromosome. By the way, chromosomes. What is this branch? What is each strand called?
Chromatid.
Chromatid, sister chromatid, right? Sister chromatid. What is this called?
Centrosome.
Is it centrosome or centromere?
Centromere.
See, it's all your fault. You kind of put me in doubt. I thought it's a centromere, but now you say
I
centrosome.
think it's centromere.
Okay, I'm going to put centromere. If it's wrong, blame it on Kevin. Centromere. I can't be doubtful. Kevin, it's all your fault, Kevin. Next time you have to buy pizza for everyone. Okay, let's see if I can get this. I know it's, let's say, chroma. So the person who's watching is laughing at us. Hey Ali, I know what it is. Chromosomes fact sheet. Drum roll, please. Drum roll. Number 2, centromere. Ah, we messed with pizza. Centromere. Kevin is right.
Yeah.
Okay, centromere. Centromere. So what is centrosome then?
I think it's the things at the ends of, like, you know, when they draw like mitosis, they draw like this big bubble, and then at the 2 poles are the centrosomes, and then the spindle fibers form from the centromere and pull it to the. I think that's what the centrosome.
Or the centrioles then.
Those are called the centrioles, but the protein that forms them connects the spindle fiber, that's called centrosome, right? Oh, we'll get to that. So here, why don't I draw this? So if this is a cell, right, nuclear membrane starts to do this, and this is the these are the chromosomes, and these are centrioles, and then, right, and then this is called centrioles, and then what is, okay, and then they start making, not yet, but eventually they make this one. Okay, dissolves, nuclear envelope dissolves. What else? What is the longest phase in mitosis? Prophase, metaphase, anaphase, or telophase? So this is the one I really want you to be careful. Longest phase, and I'm going to give you a good hint, in mitosis and meiosis, in both of them. And in meiosis, I'll get to that in a moment. You'll see why it's longest with mitosis. Okay, anything, if we don't have anything, let's move on to the next one. Metaphase. So the chromosomes line up. They call it middle plate, middle cell in the middle, or cell plate. Sometimes you hear that in the middle, and then spinal, microtubule spindle, spindles form or attached to centrosomes. This is a centrosomes. Alright, metaphase, anaphase. What happens to anaphase?
They split.
Yeah, they get pulled apart by the central.
You have to tell me what splits.
The sister chromatids split away from each other because they're pulled apart by the spindle fibers.
Sister chromatids split. There is something that I want you to know is called kinetochore. What is kinetochore?
Spindle fibers.
So these are spindle fibers. This is a sister chromatid. Spindle fibers. The kinetochore is a protein that kind of attaches the spindle fiber into this. So this is where you find the kinetochore. Now, a couple of things that they ask observation. What I saw that in the question. I never thought they would ask you this time. What happens at what stage in cell division the length of spindle fiber becomes shorter? At what stage in cell division the length of spindle fiber becomes shorter?
Becomes shorter or shortest?
Shorter.
So it starts getting shorter.
So anaphase.
Anaphase. Do you see the division, the thought? So if this is a metaphase, if they're all in the center, right? They're attached to this, and then once you go to anaphase, they start pulling away to this. And what kinetochore does, it kind of eats itself. It's something that as it pulls the sister chromatid toward itself, it just gets rid of the spindle fiber, and that's how they make it shorter. So anaphase sister chromatids and shorter spindle fibers. Again, what is spindle fibers is made of? Microtubule. I want you to make a distinction because there's one to come up. The next one is called telophase. What happens in telophase? The cell's already migrated toward the cell, and you start, excuse me, this is called cleavage. Furo. And these are the cells. Question. When this starts forming this one, they start creating this cell plate to separate them. What is this made of? Is this microtubule or is actin filament? You should know this. This is an actin filament. I saw that in one of the questions last night. When you have a telophase, when this is start to cleaving the cells, separating them, the stuff that make this separation or this wall is made of actin filament. This is the part that they add. I said I erased it. I guess prophase, metaphase, prophase, prometaphase, metaphase, anaphase, telophase, and at the end sometimes they add another stage. It's called cytokinesis. So what is cytokinesis?
Just the breaking.
Cytoplasm splits apart.
It's a stage that you just separate them. The cell division happens. You separate the cytoplasm at the end of mitosis. Are you okay for this? I went over this a bit fast because not a lot going on, but when I get to meiosis, I really want to slow down and ask questions, make a comparison. Does anybody want to add anything else? We're good? All right. So let's go over the more challenging one. And so few highlights. The longest one is prophase. So you're talking about prometaphase. What is prometaphase? Prophase is the part that you see. Question. At which stage spindle fiber form or appear?
Probably prometa, because it's between. Should be between them.
The only time some teachers, some books, they mention a prometaphase is the emphasis they want to make on spindle fiber. In prophase, just you see chromatin. Everything condenses, you see chromatin pops out. In metaphase, they are lined up. So somewhere in between prophase and metaphase or prometaphase is when you see the spindle fiber attaching to centromere, but they're kind of pulling and pushing chromosome, but they're not straight. But when it gets to metaphase is when they are straight up, right? So pro-metaphase, if you see a question, one of the major events happens in pro-metaphase that spindle fiber forms. You can see them, they're attached to chromosome, but they haven't made that adjustment, putting them lining up in the middle. All right, going to the next. This is easier. Just to save time, can you guys find mitosis in this? And there is a picture I want to see, the place that they talk about mitosis and meiosis. All right, Vivian, tell me about meiosis. What kind of division is it? Sexual. What happens to chromosome number?
It divides in half.
Right? Very good. Divides in half, becomes n. From one cell, how many cells do you make? 4 cells. So one cell becomes 4 cells. Sexual division happens in reproductive system. This is ovaries. Okay, consists of 2 cell cycle back to back. Okay, let's start with meiosis 1. So prophase. Hey, bravo. Love that. Let me come up with the one about the practice. Okay, I really want you, before I go over this, just okay. So this is 2n is equal 6. Thank you. 2n is equal 6, right? 2n is equal 6. I want you to just pay attention to how they line up. So this is a chromosome replication. So this happens in S phase. Make it in S phase, bring it in prophase, you get this one. It appears to me the red, let's say, is for female, male, I mean mom and dad. But then when it comes to the metaphase mitosis, there is no pairing of what we eventually I'm going to talk about, homologous chromosomes. So these two, the bottom one, let me borrow this. These two could be homologous chromosome, right? Let's say this is chromosome 2 and 21 from 20 from mom, this is 20 from dad, right? 18, 17, what have you. In mitosis, they do not line up. That's how they really draw them out, right? Pay attention if there's a test question, they really draw it out. But compare this in meiosis, they are the same thing, 2n is equal 6, right? 6 pairs. But in meiosis, they kind of match up next to each other. That's what's called homologous chromosome. Eventually, this ends up 1, 2, 3, 4, 5, 6, 1, 2, 3, 4, 5, 6, whereas this one eventually becomes half. So to begin with, we are all 2n, right? Everybody in our body is 2n. Skin, you make 2n, another 2n. Eyes, 2n, another 2n. But when it comes to reproductive system, you have to go 1n. And the reason they go half is because the way they line up in metaphase 1. Notice the difference between line up. So whatever you line up here, the next is just based on this. You separate this, you separate this. But since they line up next to each other, they kind of don't separate as this one. This is a good graph, good comparison to make a distinction. Same number of chromosomes to begin with. There is a match between this, but whereas this one, there is no match, so they just stay separate, right? Okay, so that's one thing. Okay. And the next one was, no matter how hard I try, I might not be able to answer it very nicely. So, prophase 1. What happens in prophase 1? We have all these homologous chromosomes coming together. What is homologous chromosome?
They're the same, like matching number.
Okay, so homologous chromosome is, let's say, if I have 2 23 pairs, right? So this is, let's say, chromosome 15 from mom, chromosome 15 from dad, right? In this picture I showed you, you see them on both. And then, let's say, this is a chromosome 10, and this is a chromosome 10. These are called, the purple and green, they are called, if you want one of these, they're called non-sister chromatid, right? These are sister chromatid, these are non-sister chromatid. I want you to make a distinction between these two. So, homologous chromosome, identical copies, but since this is from mom, this is from dad, they have different genetic makeup. Mom has a blue eyes, dad has a brown eyes, black hair, blonde hair. So they carry the same trait, is the color of eye, color of hair, but the genotype is different. One is recessive dominant. So that's a homologous chromosomes, identical copies of gene, but from different mom and dad, right? So this is homologous chromosomes, and that's how they line up. And make a distinction between non-sister chromatid and homologous chromosome versus the two purple sister chromatid. When they form in this form, when these two come together, what do we call this structure? Tetrad. Okay? Now, in prophase 1, if homologous chromosomes line up next to each other, sometimes you end up getting this structure. They swap, right? The place they swap, right at this place, this is called chiasmata. This is a centromere, right? Centromere. But the place that they start doing the synapses or crossing is a chiasmata. And when would this take place? At prophase 1. I asked you what is the longest step in meiosis. You said prophase 1. In mitosis. In meiosis, there is a reason for that, because let's say a cell that goes through meiosis, if there is no change, right? A baby is born and they say, look, you're just completely identical to your dad, right? Everything is in you is like your dad. But some baby boys say, you know what? You have your mom ears, you have the tallness like your dad. But there is a change. If there is no exchange at prophase 1, there is no genetic diversity. Would it be beneficial to have genetic exchange or not, and why? Would it be beneficial for the next generation to have crossover or not to have crossover?
Have crossover.
Double-edged sword. Sometimes dad has a disease, and then the disease carries out to kid. So at that point, you don't want a crossover. But overall, if they are both healthy, if you have a crossover, causes genetic diversity. It's called crossover recombination. And why is that? What if in a population there is a disease? If everybody carries the same genotype over generation, if disease comes in, destroys everything. But if there is a genetic crossover, there might be some resistance to a disease. That's why it's for the survival. Think about it that way too. All right, so chiasmata, where the cross happens in prophase 1. This is the longest. The cell stays at this stage as long as it can for the hope of some change. And also called recombination. You do a recombination. The other thing I want you to think about the linkage gene, linked gene. If the genes are closer to each other, let's say there are two genes, let's say A and B, the closest they are to each other, the chances of genetic recombination is high or low. So if I have two genes here, A and B, whereas the gene that is between C, I believe if they are closer to each other, I get chances is higher because there is a possibility that they swap. The possibility for them to swap is not. We'll get to that when it comes to Harvey-Weinberg equation. All right, so prophase 1, again, let me go over this. So we got homologous chromosomes. Homologous chromosome line up. And in this one, yes, I see crossing over. Notice this is blue-red, the prophase 1, and then centrosome, right? we talk about the centrosome with centriole. So centrosomes are protein happens in centriole. Centromere, as he said, is a protein, is where the two sister chromatid cross. You go to metaphase 1, notice in this case they don't say prometaphase. Had they said prometaphase, spindle would have formed prometaphase. Homologous chromosome line up, metaphase, they come up in the center. The great distinction, the question they ask over and over again, anaphase 1 and anaphase 2. Between these two, what happens in anaphase 1?
Homologous chromosomes separate.
Would sister chromatids separate in anaphase 1? Would you see any change in centromere in anaphase 1? This is the type of question they ask. Would anything happen to the centromere in anaphase 1? No, because centromere is when you attach the two chromosomes, right? If you homologous chromosomes, they're not the only thing that brings homologous chromosome together, force of adhesion or the chiasmata that they change. Otherwise, it's just through the cell cycle they come close to each other. There is no connection, there is no link, no protein, nothing. So in anaphase 1, you do not touch the centromere, it's intact. You go to anaphase 1, you separate homologous chromosomes. Next question, if this is 2N, what would this be, N or 2N? 2N. This would be N. The second meiosis, so think of it this way, what was mitosis? One cell becomes two cells, right? Would genetic number change in mitosis? 2N is 2N. So if this one wants to become two identical two cells, the way it's written probably could be counterintuitive, but whatever you make, reduction happens at the first stage. So meiosis 1 to the end of at the end of meiosis 1, from 2N you go to N. Meiosis 2 is just making one N into two N's, right? One N gets into two N's. So the first one